everyone, my name is Lauren, and today I'm going to be telling you about some of the molecular mechanisms behind the development of animals, which, as you just heard from Jack, is a great illustration of how emergence is at work in many of the processes of biological life. Even if we break life down into its simplest components, into cells, these cells have to do some pretty complicated behaviors, like consuming energy to grow, reacting to their environment, and replicating, which they do by dividing into what we call daughter cells. Cells can do all of these behaviors and many, many more using little molecular machines called proteins. Each protein in a cell has one very specific function, but they all work together to help the cell accomplish its wide variety of functions that it needs to do throughout its life. If we move now to thinking about a larger organism like a human being, this picture, of course, becomes even more complicated. One reason that a human is more complicated than, for example, a single-cell bacterium is that we contain many different kinds of cells, like the nerve cells in our brain, our muscle cells, our blood cells, and our intestine cells. It's actually estimated that humans have more than 800 distinct types of cells in our bodies. And these cells are all different from each other and can do different jobs within our bodies, largely because they contain different kinds of proteins, like the electricity sensing proteins that may be contained in a nerve cell versus the contractile proteins that are in muscle cells, versus oxygen carrying proteins in red blood cells, versus digestive proteins in intestine cells. And you can see the pattern here. Of course, then, within our bodies, these cells are organized into structures that we call tissues, like the nerve cells being organized into nervous tissues, which are then organized into even bigger structures called organs. Clearly, there's a lot of complexity here, right? And yet, incredibly, all of this complexity originally comes from just a single cell, the embryo, which is the result of the fertilization of an egg by a sperm. Here I'm showing a video of the development of a frog embryo from that single cell stage. And you can see that over the course of minutes or hours, of course this video is a little bit sped up, this single cell embryo starts to undergo divisions into many, many daughter cells. And importantly at this stage, the whole embryo isn't really growing in size, it's just distributing its contents among these daughter cells. At a certain point, of course, we can start to see more complicated behaviors emerging, like cells moving around and starting to create structures like this folding structure, which is eventually going to result in the development of the spinal cord of the frog. We can also start to see some facial features and some features that we can recognize in the adult organism. Although, of course, in the case of a frog, they have to undergo some extra developmental steps that we as humans don't do, like becoming tadpoles before becoming adult frogs. But still, we can use organisms like frogs to start to understand some of the developmental processes that also apply to humans. Now, I think it was relatively easy to see, just from this video, how some of the later stages in development have a lot of complexity behind them. But what I really want to focus on today is how even those very earliest stages of development have complex patterns emerging from some relatively simple rules. So if we focus just on that one single cell embryo and think about how it starts dividing in those first couple of hours into daughter cells, even though we couldn't see it in that video, already at this stage some of these cells are already different from each other. So one of these cells is already destined to become the head, and another is already destined to become the tail. And as the embryo continues dividing, more and more of these cells are taking on different characteristics from one another. And we're getting different populations that have distinct functions from one another. We can think of this as these cells making decisions about their fate. So they're going down a specific lineage and kind of narrowing down on the type of cell that they're eventually going to become in the adult organism. Another way to say this is that these cells are decreasing in their potential. So while the single cell organism or the single cell embryo has a huge amount of potential, it will give rise to every single cell in the adult body. As these cells make cell fate decisions, the repertoire of cells that they can give rise to becomes narrower and narrower. But importantly, at the same time, they're becoming more and more specialized in their functions. To talk about some of the molecular mechanisms behind how these cell fate decisions are made, I'm going to focus on just one cell fate decision that has been used as a model in the developmental biology field for a long time to understand these processes. And importantly, it's a cell fate decision that has to be made for every kind of organism, from humans to frogs to another great model organism that we use in developmental biology, fruit flies. And this is the differentiation between the head and the tail. And today I'm going to be focusing on some research that has been done in the fruit fly to understand how the head and the tail become different from each other in very early embryo development. So if we return back to that image of the single-cell embryo and think of it as essentially a bag full of proteins, you can imagine that as this embryo starts dividing, it's going to be dividing those proteins up into the daughter cells. And if those proteins were evenly distributed throughout the single-cell embryo, these daughters would be roughly inheriting roughly equal amounts of all of these proteins, which is fine, except that if that were true, all of these cells would be functionally the same as each other. But we know from experiments that some of these cells are already different from the others. Some of them are already destined to become the tail. So how does that happen? How are some of these cells becoming different from the other ones? It turns out that the key is that these proteins were not evenly distributed within the embryo in the first place. In particular, there's one really key protein, which I'm representing with this dark blue horseshoe kind of shape, which is distributed into one particular section of the embryo. And its function is to bind to and essentially trap all of the tail-producing factors. So at this stage, these proteins aren't really stuck in one place in the cell. They're all moving around. And as this trapping protein interacts with the tail-producing factors, they become concentrated there. They're trapped there. And so then once that embryo starts dividing, these daughter cells are not actually inheriting those tail-producing proteins equally. Only a subset of the daughter cells have these tail-producing proteins or tail-encoding proteins. And so only that subset of daughter cells is eventually going to become the tail of the fly, or it's taken on this tail cell fate decision. And all of this really comes down to that really quite simple rule of one protein being asymmetrically distributed within the single-cell embryo. And even though this is just one cell fate decision that I've told you about so far, it turns out that, especially in the fruit fly, the same general principle and mechanism applies to many different cell fate decisions in the early fly embryo. Here I'm showing a representation of a fruit fly embryo at a somewhat later stage. You can see that there are many, many more cells, and they're color-coded based on the cell fate decisions that they've taken, or the protein composition that they have. And even this is an oversimplification of the patterns that are already present at this stage. But we can see some really interesting things, like that head versus tail differentiation, and also this really striking banding pattern, which corresponds to the development of precisely 8 segments in the adult fly's body. All of this incredible pattern is emerging from those relatively simple rules of protein-protein interactions and protein distributions within the embryo. So that's one of the major mechanisms by which we can get from a single-cell embryo to a complicated organism like a human or a fly. But it's also important to remember that we don't stop developing once we're all grown up. In particular, at the cellular level, there are many different kinds of cells and tissues that need to be continually replenished throughout our lives. In particular, I think it's pretty easy to imagine how or why the blood would need to be replenished frequently in our lives, whether it's because we're losing blood due to bleeding, or doing blood tests at the doctor, or just natural cycles of death and replenishment of our blood. In fact, every couple of months we have completely new blood cells in our system. The blood is actually made up of many different kinds of cells, and this is just a subset of them. And they all have different jobs related to transporting oxygen around the body, the immune system, blood clotting, etc. And yet, incredibly, all of these different kinds of cells are replenished by a single type of cell, a blood stem cell. And the molecular mechanisms by which this blood stem cell, this one type of cell, can lead to the wide variety of different kinds of cells in the mature blood has a lot of parallels with the mechanisms that we just learned about for embryo development. But before getting into that, I want to just take a moment to talk about what exactly a stem cell is. So there are really two important components of the definition of a stem cell. The first is, as we've just seen, that a stem cell can produce multiple different kinds of cells. This is a process called differentiation, because the stem cell is producing cells that are different from itself and also different from each other. The other really important part of a stem cell's identity is that it can produce new stem cells, which we call self-renewal. And this is really key because if the stem cell could only differentiate, could only produce those mature, let's say, blood cells, we would quickly just lose all the stem cells and not be able to replenish, for example, our blood for the rest of our lives. So both of these functions are really, really key to the identity of a stem cell. And in the case of a blood stem cell especially, because it can give rise to so many different kinds of mature cells, there have to be many of these different differentiation steps. So a stem cell's daughter will follow a certain path or lineage of differentiation, which you can also think of as those cells making cell fate choices, where they're becoming more and more specialized in their function, but also, in a sense, losing potential in terms of how many different kinds of cells they can give rise to. So focusing back on that initial decision that the stem cell has to make, how does it know whether to differentiate or to self-renew? There's been a huge amount of research on this question over the last couple of decades, and it turns out that it has everything to do with where a stem cell lives in the body. Every stem cell has its own particular niche, which is basically just a microenvironment in the tissue that it resides in. And in the case of the blood stem cell, this niche is in the bone marrow. And the function of the niche is to essentially provide the stem cell with signals about what kind of cell fate decisions it should be making. And usually the niche is really giving the stem cell signals to keep self-renewing and not to differentiate. And so long as the stem cell stays nice and tucked up in the niche, it's getting lots of these self-renewal signals, it never will differentiate. But if the stem cell divides such that the daughter cell moves a little bit farther away from the niche, it's now getting fewer and fewer of these self-renewal factors. And just because it's gone below that threshold of signal factors, it ends up differentiating instead of continuing to self-renew. And now that's just the case of the original blood stem cell deciding whether or not to make that initial differentiation decision. But as I just showed you, the stem cell differentiation pathway actually includes many different steps of differentiation. And we don't yet know all of the signals that tell these daughter cells how to differentiate at all of these different steps. But it seems like this similar principle applies to all of them, where the particular location within the bone marrow or the blood vessel that a cell finds itself in makes it able to receive different kinds of signals that instruct it about what kinds of cell fate decisions to be making. All of this is an incredible example of emergence, where we again have that very simple principle of just protein concentrations in different areas of the tissue, leading to the development of a pretty complex and thankfully very functional tissue, the blood. So yes, life is really complicated, but it's not so complicated that we can't understand it as scientists. We have been able to break it down into some relatively simple processes that we can understand. Like in the case of the single-cell embryo or the blood stem cell, where we've been able to find that the relatively simple rule of protein distribution leading to cell fate choices is really key to the emergence of complexity in both of those systems. I also really want to emphasize that in both of these systems, it would be foolish to try to reduce them down too far, to take that reductionist approach. So you can imagine, for example, if we had tried to study the function of each of these individual proteins completely in isolation from each other, we wouldn't have been able to get very far in understanding these developmental processes. Because as you've seen, the keys here really have to do with protein-protein interactions or cell-cell interactions. That's really what leads to the emergence of all this complexity. So now that Jack and I have told you about how emergence is at work as proteins build cells and then cells build organisms, next we're going to hear from Alyssa about how emergence is also at work in the way that organisms organize themselves into collectives and communities. Thank you.